So at the outset of Lecture 4, we would typically start with some discussion of the War on Science video that I had you watch. Think about the facts of the case as opposed to the drama. I want you to look past the strong personalities that were there. What are the central issues? To start with, what's the difference between the traditional old-style creationism as it was taught in the 1960s? If you remember, this is what the Dover School Board originally wanted to bring back, versus the newer intelligent design or ID creationism as was carefully crafted by Johnson, Meyer and company. When I watch this video, I see a boundary that was crossed when the argument shifted from what started as there is no known evolutionary mechanism that can account for this, but then ended up as some greater force with intelligence must be responsible for this. The first statement is owning up to a gap in our ability to explain something. The second is proposing an actual explanation, a supernatural one. ID creationists think that the first statement is actually evidence in support of the second, but in order to think this, you'd have to have a very poor understanding of the nature of science and the nature of evidence. Admitting one's ignorance is a part of science, and so the first statement is perfectly fine. There's nothing inherently wrong with, for example, saying that the bacterial flagellum cannot be explained by any known mechanism of evolution, though Ken Miller actually does a good job of demonstrating that a stepwise path to the flagellum through gradually increasing complexity is actually possible. He didn't have to do this, though. If not this, then there are plenty of things for which we lack adequate explanations. And in fact, this is what makes science interesting. Again, nothing wrong with the first statement. It's the second statement where ID claims that the supernatural explanation is somehow scientifically supported by the first statement. This is where intelligent design fails completely. We've already pointed out a couple of times that science requires at least the possibility of validation through experimentation and observations from the natural universe. And by definition, any kind of supernatural intelligence is outside of this domain. Those of us who have been engaged in science education are familiar with a general rule known by the acronym NOMA, short for Non-Overlapping Magisteria. This mouthful of a term was coined by the late Stephen Jay Gould, a Harvard paleontologist and biologist who was instrumental in promoting evolutionary science in the late 1900s. One of his many campaigns was directed at reducing the friction between evolutionary biology and religion. After Gould's meeting with Pope John Paul II, the Roman Catholic Church officially changed its doctrine, specifically with its full acceptance of the validity of Darwin's theory. And the principle of non-overlapping magisteria was the basis for this change. Like many negotiated peace agreements, NOMA removes conflict by separating the two domains, or magisteria. Here you have the magisterium of science and the magisterium of things beyond the natural universe. The magisterium of science is the domain of all things governed by the natural laws. Here is where explanations are subject to validation by supporting evidence or to refutal by compelling counter-evidence. Everything we discuss within the confines of our science classroom will be part of this magisterium. The other magisterium is not one that I can offer much authority on personally, but presumably religion and all forms of belief that do not require evidence or validation belong there. Gould says that as long as we stay within our zip code and they stay within theirs, nobody gets hurt and we can all carry on with the work at hand. As long as these magisteria remain separate and non-overlapping, we're good. And clearly, intelligent design creationism is an attempt from the other domain to poach within our magisterium of science. Not good. If, in the context of a science class, I were to talk about spirituality or soul, whatever that is, this would also be not good. I'd be doing the same kind of poaching that the ID creationists are attempting. So don't worry, there's not much risk of this happening, although we on the science side of things still need to be careful about maintaining objectivity, specifically where it comes to powerful arguments like evolution by natural selection. What I'm saying is that it's easy to fall into a trap of presuming that natural selection and adaptation are what made something the way it is, even when natural selection and adaptation may actually have had nothing to do with it. I'll give you an example. Some of you might be aware of the normal shape of our red blood cells. They're biconcave discs, kind of like a donut where the hole doesn't go all the way through, or a hockey puck with indentations in the middle of both of its flat faces. If you think about the significance of this shape and why it is biconcave instead, for example, of being a regular flat-faced hockey puck, you might note that with this biconcave shape, there is a greater surface area on the cell relative to a regular hockey puck. The cell's ability to take up oxygen and release oxygen benefits from having this little bit of extra surface area. And so you might conclude that this biconcave shape is the product of natural selection that had the result of making red blood cells better suited to the role of loading and unloading oxygen in the animal's bloodstream. Now, this may seem perfectly reasonable, and lacking any better explanation, it might be even acceptable. But in the case of red blood cells, I could explain this perfectly well without natural selection for altered cell shape. Part of the differentiation of these highly specialized cells involves the removal of the cell's nucleus. There is no nucleus in mammalian red blood cells. They don't need them because they won't ever divide again, and all the transcription of all the RNA they'll ever need is done. So just before a red blood cell is finished and declared ready to be put into circulation, the nucleus is ejected. These ejected nuclei are recovered and the materials are put to use in the biosynthesis of more cells. This is thought to be more efficient than having the nuclei circulate with the cells, although enucleation is something that only occurs in mammals. All the other animals leave the nuclei in the red blood cells and they do perfectly fine. Now, the enucleation of the developing red blood cells leaves a void in the center of an otherwise disc-shaped cytoskeleton. And that space, once occupied by the nucleus, is never filled back with other stuff. The plasma membrane has to pucker inward on both sides, and the result is a biconcave shape. So, is the biconcave shape a product of evolution? Yes, of course it is. Whatever the evolutionary force causing enucleation in the first place resulted in this shape. But is it the product of natural selection favoring an increase in surface area of the cell? Well, I suppose it's possible that this might have contributed, but given that the enucleation alone is sufficient to account for the cell shape, why would we attribute its evolution to anything else? The explanation based on natural selection may have come to mind because we are so conditioned to presume that everything in biology is the way it is because of evolution by natural selection. But in this particular case, giving credit to natural selection would not be founded upon anything we could gather from the natural universe. It may not be as blatant a violation of NOMA as if I, as science teacher, were to expound on spirituality or soul. No, not quite at that level. But this tendency to attribute everything to the Darwinian mechanism borders on an act of faith by not demanding validation from supporting evidence. And so, in a way, this also violates the rule of non-overlapping magisteria. And this is problematic for us because it gives the other side a valid reason to kvetch. I'm going to ask you to be mindful of our responsibilities as workers within the magisterium of science. There are a few other related traps that lie in the way, and I want to follow up with one that's also one of the most problematic: teleological thinking in evolution. The word teleology comes from the Greek telos, which means ultimate objective or aim. Any activity that is directed toward a particular goal is teleological. Most of everything we do while we're conscious and awake is directed at a particular goal. My walking to the kitchen to get a glass of water because I'm thirsty, that's teleological. If you can recall the last 500 things you did between waking up and starting this lesson, I'd bet that most, if not all of them, are teleological. I maintain that we're conditioned to presume that things happen naturally via preordained teleological paths because that's the way things unfold in our conscious lives. But in evolution, there is no end goal. The finches in the Galapagos didn't have a plan to evolve any particular beak shape because that's what would be best for their ability to get food. The process of natural selection may have had the outcome of the beaks evolving into an adaptive morphology, but this occurred without any forethought, without any predetermined goal. I would hope that by now you're able to explain how evolution by natural selection is a process causing incremental microscale changes favoring the most successful phenotypes in any particular generation. Over thousands of generations, this gives rise to larger scale adaptive change, and at some level, this might seem like it was the objective all along. But of course, there really never was a goal at all, right? I'm saying this because I expect that you're there nodding your head and thinking how silly it would be for anyone to apply that evolutionary change is directed by some preordained goal. And yet, we've all seen the graphics in which there's a procession of animals, starting on one end with a fish emerging from a lake and going through a sequence of salamander, lizard, rat, monkey, ape, caveman, and ending with a modern human, all of which are walking in the same general direction from water onto land. We have heard terms like advanced forms of life and evolutionary ladder and evolutionary progress used, even by biologists who really should know better. Now, what is wrong with these concepts? Now, it's your job in this class to attain clear, precise understanding of concepts and to communicate this in clear, precise language. If a student writes in their essay that pigs are a more advanced form of life relative to insects, to me, this demonstrates a fundamentally flawed understanding of biology. This student appears stuck on some view in which there's a preordained direction for evolution. The word advanced is the problem. After all, you can't be more or less advanced unless there's a particular place that you're headed. There's one more thing about NOMA. It's kind of deep, kind of serious. Remember that these two magisteria are supposed to be non-overlapping? Well, what happens if there is some overlap? I ask this because I'm old enough to remember back to the Obama administration when Francis Collins was selected to serve as the director of the National Institutes of Health, the NIH. Collins is a brilliant scientist and even more brilliant as a scientific manager. His leadership is a big part of the reason that the Human Genome Project, the sequencing of the complete human genome, was completed years ahead of schedule. In your millennial mindset, you might think that we've always been able to get a full read of our genomes for less than $1,000, but no. The first human genome was a project costing billions of dollars and requiring the efforts of teams of gene sequencing hotshots across the globe. Each team did a little piece that was independently validated by another team. In coordinating this huge complex effort, Collins was definitely the man. So obviously, he was a great choice to be the NIH director, right? Well, yes. In retrospect, it seems he was just the right choice. It's no small achievement that he is one of a very small number of Obama appointees that continue to work in the same capacity with the blessing of the Trump administration. But at the time of his appointment, there were some concerns. Collins, an evangelical Christian, had been on record enumerating some of the things that he felt were outside the domain of science, things that could never be elucidated without appeal to a greater intelligence. Basically, he was being a good adherent of NOMA, and he was evidently very good at this, given that he had clearly found peace between his scientific work and his faith. Here are the things that belong in the science basket, and here are the things that belong in the other basket. Collins was telling us that you don't have to give up religion to be successful as a scientist. But among the things that Collins put in the other basket was an understanding of human consciousness. This, Collins wrote, was outside the domain of what could be known through science. Two things to note here. One, there are a lot of neuroscientists and behavioral scientists whose research on the human brain address specifically things relating to consciousness. Two, the National Institutes of Health is one of the main conduits for supporting such research through federal resources. Oh, how the alarm bells rang. Would Collins be pressuring to cut off funding for certain kinds of research because he felt they were outside the magisterium of science? Again, in retrospect, this turned out to be a non-issue. Collins has fully supported neuroscience and behavioral science as well as anyone could have hoped. The American Brain Initiative and the Human Connectome Project are two mega projects spearheaded by the NIH. If you're interested at all in neuroscience, I strongly suggest that you look into these. My point here is that we tend to go about our lives in science following the rules of NOMA pretty uneventfully. And then wham, all of a sudden some issue arises. A school board in Pennsylvania gets taken over by creationists, or maybe there's a questionable appointment to national science leadership, and boom, we're thinking seriously about NOMA again. Okay, enough on the documentary video. Let's get back to our 4 microevolutionary forces. At the end of the last lecture, I introduced the notion of a theoretical non-evolving population, subject to none of the microevolutionary forces and undergoing completely random mating. The Hardy-Weinberg law says that in such a population, the frequencies of the genotypes will be as follows. The genotype frequency of the big A homozygote will be P, that's the frequency of big A, squared. The genotype frequency of the little a homozygote will be Q squared. And the genotype frequency of the heterozygote, big A little a, will be 2pq. These are the frequencies of the 3 genotypes in a population with allele frequency of big A equal to P and the frequency of little a equal to Q if the population is non-evolving and random mating. What this does for us is it gives us a standard to compare against. Take our Snapdragon population from the previous lecture with genotype frequencies 0.6, 0.3, and 0.1 for big A, little a, and little a. We calculated P for this population to be 0.75 and Q to be 0.25. With these values of P and Q, the Hardy-Weinberg law states that a non-evolving population will have genotype frequencies of big A at P squared or 0.75 squared or 0.5625, big A little a at 2p cubed or 2 times 0.75 times 0.25 or 0.375, and little a at 0.25 squared or 0.0625. I'll use the caret symbol or hat above the G. You can say G hat to distinguish the Hardy-Weinberg theoretical genotype frequencies from the genotype frequencies that we calculated for our actual population. Remember that the G hats do not come from a real population. It's the theoretical population that has random mating and is subject to 0 evolutionary forces. On the other hand, These over here are the genotype frequencies we actually have in our real population, as calculated from the numbers of organisms that we're given. So our thinking is this: if my population were actually not evolving, then it would have to look like this. If my population does not look like a non-evolving population, then it must be evolving. The only possible causes of evolution are natural selection, random genetic drift, mutation, and gene flow. Therefore, if my population is different from the Hardy-Weinberg expectations for a non-evolving population, then one or more of these evolutionary forces must be acting. There is, however, one other possibility for a departure from Hardy-Weinberg, and this would be if mating is non-random. Remember that random mating is a condition for the Hardy-Weinberg law. It's for this reason that we can add non-random mating as a fifth microevolutionary force, though it's really more like a fifth potential cause for a population to be different from the Hardy-Weinberg genotype frequencies. So our Snapdragon population had genotype frequencies of 0.6, 0.3, and 0.1. The theoretical non-evolving population has genotype frequencies of 0.5625, 0.375, and 0.0625. These populations are different. The numbers do not match up. And therefore, we conclude that our population is evolving through one or more of the microevolutionary forces and or through non-random mating. If you find that a population departs from Hardy-Weinberg expectations, you can also characterize this departure in one of two ways. Either there will be an excess of both homozygotes and a shortage of heterozygotes, or there'll be an excess of heterozygotes and a shortage of both homozygotes. It has to be one of these two. If you're only looking at a snapshot in time, you can't tell if there's selection favoring one or the other homozygote, because that would have to result in a change in the allele frequencies, P and Q. In this analysis, we have the same P and Q for both the actual population and the Hardy-Weinberg theoretical non-evolving population. Recall that we used the actual population to calculate the allele frequencies, and then we used these same allele frequencies to produce the Hardy-Weinberg genotype frequencies. In this Snapdragon example, we determined that P equal to 0.75 and Q equal to 0.25 from the actual population and used these same numbers to calculate Hardy-Weinberg. So what is the status of our Snapdragon population? Is there an excess of homozygotes or an excess of heterozygotes? We're about at the halfway point here, so let's go grab a snack or something. I'll give you the answer after the break.